Good morning. Thanks for joining us for Informa Pharma Intelligence's ASCO Essentials webinar. My name is Mary Jo Laffler. I am the executive editor for US and commercial and R&D news for Scrip, the Pink Sheet, and In Vivo. I'm joined here today by senior analysts from across the Informa Pharma Intelligence products, and I'd like to ask everyone to introduce themselves.
Hi, my name is Maria Birizina, and I'm an oncology analyst at TrialTrove.
Hi, I'm David Dehan, and I'm a senior analyst at Biomedtracker.
And I'm Zach McClellan, an oncology analyst with Datamonitor Healthcare.
Thanks, everyone. Well, the first week of June is fast approaching, and planning for the American Society for Clinical Oncology Annual Meeting is in full swing. This year's meeting has a jam-packed agenda as usual, and our analyst team has been working long hours to sort through the hundreds of abstracts and help identify what you need to know. Informa Pharma Intelligence has already released a pre-meeting planner with a more comprehensive look at the abstract releases. During the conference, our analysts on the ground will put out a mid-meeting report, and then stay tuned for a post-meeting report coming out. In addition, Scrip and Pink Sheet will be publishing reports before, during, and after ASCO, both by reporters in Chicago at the meeting and by reporters around the world. And you can follow our analyst and journalist teams on Twitter for commentary and data throughout the meeting. We'll send out an email with this information so you can click through to whatever you'd like to access at the end of the call. So let's jump into things. Immuno-oncology remains front and center, and we're going to start things out with a look at some of the new checkpoint modulators coming down the pipeline, as well as other emerging IO modalities like TLR9 and CAR-T. Then we'll review how the ASCO data looks for the IO leaders like Merck's Keytruda and Bristol Myers Squibb's Opdivo in the all-important first-line lung cancer setting. We'll also look at a potential targeted therapy upset in first-line non-small cell lung cancer with Pfizer's Datamonitor. Then we'll shift focus to look at new developments in breast cancer. If there's time, we'll take questions at the end, but please do use the chat function to submit a question at any time. We'll review the questions later and respond by email if we don't get to them during the call. So, Maria, there's been a lot of disappointment with checkpoint inhibitors beyond PD-1, PD-L1, and CTLA-4, most recently with the IDO inhibitors. What should we be tracking now, and how is that data looking?
Hi. Thank you, Mary Jo. Yeah, a lot is happening in IO space. This will be an interesting year for immunotherapies at ASCO. Unlike last year's ASCO, I think this year there happened to be more clinical updates for unapproved checkpoint modulators, and so I wanted to start by talking about that a little bit. As we all know, the checkpoint modulator space is expanding. To capture the current state of the space, I made this graph, which is shown on the left side of the current slide. It shows top 15 checkpoint targets in active development. For each target, the bar shows the number of unique drugs in development, and the counts are given for the number of drugs in different stages of development. So for the PD-1 bar, there are 15 unique drugs in phase 1 development, 4 in phase 3, 5 in phase 5 and phase 3, and so on. So obviously PD-1 and PD-L1s are dominating the space, but there is a diverse next wave of checkpoints in development, and this next wave has quite a few targets, and therapies in later stages development include drugs that target IDO, LAG-3, ICAS, GITR, among others. I think that's kind of what constitutes the next wave that's up and coming. Now, on the right-hand side, I have a table that shows some of the new therapies that will have updated or first data presented at ASCO. This is not an exhaustive table, but it's kind of meant to highlight abstracts and drugs that are interesting and I think are worth the attention during this year's meeting. In checkpoint modulator space, Merck will present its first data for anti-GITR antibody, MK1248. and this is one of the abstracts that I will talk about in just a little bit. Also, Jones will present updated data from its ICONIC trial, and this is which is a study that looks at their ICOS agonist JTX 2011, and I will talk about this one also. So these two have little stars next to them because I'm going to talk about them in more detail. Another things to note is Cytomax will be presenting updated data for their PD-L1 Probody as monotherapy or in combination with ipilimumab. There will be multiple updates from Merck for their bifunctional fusion protein, M7828. I wanted to note that one of the abstracts for this compound, specifically, it will be poster 9017. It will be updated results for an expansion cohort in non-small cell lung cancer. The data looks interesting, and I encourage anyone following lung cancer and new checkpoints to check out that poster. There are a few other abstracts on this table. I wanted to know Agenus will be presented first data for their PD-1 antibody, and Seattle Genetics will also be presented first data for their CD40 antibody. There are a few non-checkpoint immunotherapies that I thought were interesting, and they're listed in the bottom half of this table. My colleague David will talk about SD101 from Dynavax later in this meeting. So overall, I built this as a reference and an overview for anyone looking at checkpoints updates at ASCO. You can take a screenshot of this or email us, and we'll be happy to share the slide. So that's kind of an overview and a summary. Now I want to talk about the ICONIC trial from Johns. So ICONIC trial looked at JTX 2011 with or without nivolumab. The study had a phase 1 run-in and then several disease-specific cohorts. The ASCO abstracts focused on gastric and TNBC cohorts. This slide shows a table summarizing responses in different treatment arms. So there were monotherapy arms as well as for two disease cohorts as well as combination cohorts. Overall, these data responses are kind of negative, and this data had was interpreted negatively and resulted in Jones's stock price going down after the abstracts were released. What I wanted to do here is actually highlight why the data was interpreted negatively, but also give some context and maybe provide a different way of looking at it, a more positive way. So the first thing to note here is that the monotherapy results in the study agree with preclinical data for ICOS agonists. ICOS is upregulated with concurrent blockade of another checkpoint, namely PD-1 or CTLA-4, so we don't expect ICOS to be efficacious as monotherapy. What was disappointing is the combination treatment in gastric cancer. The data for this was fairly underwhelming. If we look at JTX 2011 in combination with nivolumab in gastric cohort, 19 patients were treated. There were only 2 partial responses and 2 stable diseases. Nivolumab as monotherapy in third-line gastric cancer has an ORR of 12% and disease control rate of 32%. So this data is from a lot fewer patients, but the ORR is 10% and disease control rate is only 21% compared to 32% of the nivolumab being as monotherapy. So the expectation was that the combination of nivolumab and ICOS agonist would have higher ORR, and that did not happen. So I think that's sort of the bulk of the sort of negative aspect of this data. Another thing here is that they had some patients with ICOS high status in tumor tissue, and that did not translate into response for these patients. So there is no preselective biomarker at this point. Now I want to be a devil's advocate here and say that perhaps PD-1 is not an optimal combination therapy for ICOS agonist. I personally find preclinical data for CTLA-4 combination more compelling, and maybe what happened here is they decided to look for combination with PD-1 blockade because it's a less toxic and more familiar treatment with physicians, and it's also sort of more prevalent combination to go for. Johns did add a combination with zolbetuximab arm to the study, and I think that data is actually, to me personally, is a little more interesting. So it's also important to note that these patients were very heavily pretreated, potentially more than 3 lines of therapy, which would make these results a little more positive as well. Last but not least, ICOS is a costimulatory checkpoint expressed on T cells and is activated by same ligand as CTLA-4. Actually, next generation CAR T therapies now have costimulatory domain, use ICOS as costimulatory domain. So it is not an environment modulator like IDO. I think disappointing IDO data is making investors perhaps more cautious of new checkpoints, but I want to argue that ICOS is a checkpoint that is directly involved in antigen presented cell to T cell interaction and is a more compelling target than IDO, which is sort of simply a metabolic enzyme involved in tumor microenvironment. So at ASCO, Jones will present updated data with later cutoff date. So with all this said, I encourage people interested in ICOS and new checkpoint targets to listen to the presentation and understand full data. So I want to encourage you to look for sort of biological activity, what are the onset of the responses that they're seeing, maybe they now will see more responses, how heavily pretreated these patients were. So this data will not be, maybe this data will not be as disappointing, and maybe this is just a bump in the developmental road for GTX 2011. So that's my 5 cents on the ICONIC trial. With that, I want to briefly talk about MK-1248, which is Merck's anti-GITR antibody. This will be a poster, abstract 3029. So the poster will present data from Merck's anti-GITR program, and it's the second anti-GITR to present clinical data for combination with PD-1 antagonist. Last year, BMS presented the data for their anti-GITR compound, BMS 986156, with or without nivolumab. What I wanted to do is compare the data for MK-1248 presented in the abstract with the data that was presented for the BMS GITR compound. On the slide, you see a table comparing the two trials. First thing to note is that for Merck's drugs, reports data from fewer patients. In terms of tumor types, both trials were standard all-comers trials with large number of different tumor types. The interesting thing to note is the difference in adverse event profile for two drugs. BMS GITR therapy had fever, chills, and fatigue as most common AEs, and most were grade 1 or 2. On the other hand, the abstract for MK-1248 lists number of gastrointestinal events, and vomiting is listed as the first one. I would assume that there may be listed as most common to least common. I don't know. I would need to see the poster, but there are quite a few gastrointestinal symptoms that are listed in the abstract. Note that there was no vomiting listed for the BMS trial in their presentation, and nausea was very infrequent. Overall, Merck's trial had close to 100% of patients experiencing an adverse event, while BMS trial only had about 70. Furthermore, the abstract for Merck's GITR is reporting close to 50% rate of adverse events that were grade 3 or higher, but that rate seems to have been only 6% for the BMS trial. So the safety profile of Merck's drug appears to be different, and there appears to be a higher incidence of grade 3 or 4 events. The efficacy of the two drug combination regimens appears to be similar, and it's interesting to see that there is one complete response in the Merck's trial. Things to look for at the poster during the presentation, during the meeting, is biological activity, so proliferation, activation of CD8 positive T cells, NK cells, and safety profile. In early studies, that's kind of what you want to look for, other than early efficacy signals. The bottom line here is that I think GITR is a very interesting up-and-coming checkpoint target. The early efficacy signals of the combination regimens are promising. I want to note that some of the patients that responded to combination arm in BMS trial were previously treated with PD-1 blockade and have progressed on it, which supports the idea of GITR plus PD-1 blockade maybe being a rescue or follow-up regimens for patients progressing on PD-1 blockade. The difference in safety profiles is intriguing. I personally look forward to learning more about MK-1248 at the meeting. I'm very excited about this. So that's my kind of all I wanted to mention about this abstract and checkpoints.
Thanks, Maria. Very comprehensive, a lot of exciting things to keep an eye on. As you noted, there's a few more things we wanted to look into in IO. And one class that's been in development for a long time is the TLR9 agonist. David, one of your picks was Dynavax's SD101. Why is that one to watch?
All right, thanks, Mary Jo. All right, so at ASCO, Dynavax will be presenting updated data from an early phase trial evaluating SD101 combined with Keytruda in patients with advanced melanoma. The company's poster will be discussed during a poster discussion session set for Monday afternoon. SD101 is an agonist of toll-like receptor 9, which recognizes bacterial DNA and is part of the host innate immune system. It consists of a short stretch of synthetically produced DNA that is injected into the tumor. It is designed to activate plasmacytoid dendritic cells, a rare type of immune cell that secretes large quantities of type 1 interferon, changing the tumor environment from cold to hot and priming the immune system against the tumor. The trial involved melanoma patients naive to anti-PD-1 therapy, and the abstract submitted to ASCO had data for 25 patients. The 60% overall response rate and 12% complete response rate shown in the table compare well to historical data for the approved Opdivo-Yervoy combination, and that is without all the adverse events associated with Yervoy. For comparison, Opdivo-Yervoy reported a 59% overall response rate and a 22% complete response rate in its pivotal phase 2 trial. The response rates for SD101 Keytruda also compare well to Opdivo monotherapy, which reported a 45% overall response rate and 15% complete response rate. One caveat to this analysis is that these SD101 Keytruda response rates are for the per-protocol population, and adding back the 5 patients who discontinued gives an ITT overall response rate of 50% and a complete response rate of 10%, which, while not as impressive, is still respectable. We will now be looking forward to the poster at ASCO, which will present updated data from 50 patients. Other companies investigating TLR9 agonists combined with checkpoint inhibitors include Checkmate Pharmaceuticals, which presented data from its phase 1b trial at AACR. Checkmate is evaluating its compound, CMP001, combined with Keytruda in a different patient population than SD101, melanoma patients who failed Keytruda. They reported an overall response rate of 22% in 69 patients. Of course, these numbers cannot be compared to SD101 because they are completely different populations, those who failed Keytruda versus Keytruda naive. The most advanced TLR9 agonist is Idera Pharmaceuticals IMO 2125, which is currently being evaluated in a pivotal phase 3 trial in combination with Yervoy in melanoma patients who failed a PD-1 inhibitor. At ASCO, Idera will be presenting data from a phase 2 trial in a similar patient population. In their ASCO abstract, they disclosed a 47% overall response rate in 24 patients. Their poster will be on Monday and will also be discussed during Monday afternoon's poster discussion session. Back to you, Mary Jo.
Thanks, David. Seems like a very interesting class to monitor. The other IO approach that's had a lot of momentum in the last year really is chimeric antigen receptor T-cell therapy, CAR-T. Zach, could you fill us in on what advances being made in this space will be featured at ASCO?
Yeah, sure. Thanks, Mary Jo. So a Chinese research group posted an interesting question with a poster here that will be presented at Abstract 3041, presented on Monday. And they posed the question, which is better in CD19 CAR-T relapse refractory ALL patients, a CD28 or a 4-1BB co-stimulatory domain? So the trial they ran included 47 relapse refractory B-cell ALL patients, 19 in the CD28 group and 28 in the 4-1BB group. The overall ORR was 96%, which favored the 4-1BB group specifically at 100% over the CD28 group at 89%. All patients that responded did achieve MRD negativity, and peak CAR T-cell expansion favored the 4-1BB group. And notably, we don't have further information on the specific manufacturing process, but all 41 patients did receive at least one infusion of treatment and one clinical evaluation, so no patients were unable to be dosed, which is encouraging. As far as tolerability, cytokine release syndrome occurred in 45 of 47 patients, which is not to be unexpected with CAR-T treatment. Of the 5 grade 3 or greater CRS events, all were in the CD28 group. And the cytokine release peak also favored the CD28 group, which was significantly higher. 9 patients experienced all grade neurotox, and of 5 grade 3 or greater neurotox events, again, all favored the CD28 group compared to 4-1BB. So from this trial, the implication in the real-world setting is that the 4-1BB costimulatory domain appears to be more effective and better tolerated than CAR T cells using the CD28 costimulatory domain, reflecting positively for Novartis' Kymriah and potentially negatively for Gilead's Yescarta. And there seems to be some already data out there in clinical trials. One obvious caveat being this is a cross-trial comparison, which is not ideal. But previously in ALL patients, Gilead's Yescarta in the ZUMA-3 trial for adult relapsed refractory ALL patients shown a complete response rate of 71%. But again, tolerability might be a concern with grade 3 or greater CRS and neurologic events occurring in 25% and 63% of patients respectively. And resulting from that, tocilizumab and steroids were given in 94% and 75% of patients to combat those side effects. Novartis' Kymriah in ALL patients in the Alania trial, 83% of those patients achieved a complete response rate at a 3-month follow-up, so slightly better than Gilead's Yescarta. And the CRS in those patients occurred in 79% of both young and pediatric adult patients, including grade 2 or greater in 49% of patients. As far as tolerability, again, 50% of those patients, almost half of what was seen with Gilead's Yescarta, required tocilizumab, and 26% received corticosteroids. So in addition to the obvious caveat of cross-trial comparisons, the trial ran here by this Chinese research group, we need to keep in mind that full response information and detail on patient characteristics are needed to pass judgment. This is an early phase trial, but it is compelling evidence for CAR T cells manufactured under the same process. At the meeting, this is a poster, but hopefully we do have updated data, including manufacturing information about both of these CAR Ts with their different costimulatory domains, in addition to specific patient characteristics that might give us a better idea of why these different CAR T cells responded the way they did. And that's it. Back to you, Mary Jo.
Thanks, Zach. So I'm going to sort of get into a little more depth here with some of these. First-line lung cancer still represents the largest market for immuno-oncology, and there's been a lot of very focused development there, not all successful. So far, Merck's Keytruda is approved for frontline monotherapy and combination treatment with chemotherapy, but it should be joined on the market soon by competition. And a lot of that took the stage at the AACR meeting in April. David, what's left to learn about this space after such major data releases already this year?
Thanks, Mary Jo. So at AACR, Keytruda combined with chemotherapy faced off against Opdivo combined with Yervoy in a first-line non-small cell lung cancer. While the Opdivo combination reported respectable results, Keytruda was crowned the winner. At ASCO, it looks like we're going to have a face-off between Merck's Keytruda combined with chemotherapy and Genatech and Roche's Centric combined with chemotherapy in the same indication, first-line lung cancer. I'm going to start with a late-breaking abstract that will be presented on Sunday for Keytruda monotherapy. Recall that Merck's Keytruda was the first PD-1, PD-L1 inhibitor approved for first-line lung cancer and now dominates this line of therapy. It was approved as monotherapy for both squamous and non-squamous lung cancer with no EGFR or ALK genomic tumor aberrations. Only 10%, and it was approved for only 10% of patients with a high PD-L1 expression, which was defined as a tumor proportion score, or TPS, greater than or equal to 50%. The late-breaking abstract will present the first numerical data for KEYNOTE-042, a phase 3 trial evaluating Keytruda for first-line lung cancer. The trial is testing Keytruda monotherapy for both squamous and non-squamous lung cancer, again, with no EGFR or ALK genomic tumor aberrations. The big difference is that the trial is evaluating patients with a PD-L1 TPS greater than or equal to 1%. The comparator in this trial is platinum-based chemotherapy, and the primary endpoint is overall survival in patients with TPS scores of greater than 50%, 20%, and 1%. On April 9th, Merck announced that all primary endpoints were met, and the first numerical results from KEYNOTE-042 will be presented at ASCO. This data should allow Keytruda's label to be expanded from the current indication covering patients with a PD-L1 TPS of greater than 50%, and the numerical data should give us a better sense of how Keytruda performs in these different TPS populations. Switching gears now to Roche and Genatech, evaluating Centric plus chemotherapy as first-line therapy in advanced squamous non-small cell lung cancer. Back in March, Genatech and Roche announced top-line results from Empower 131. The trial is evaluating Centric in combination with carboplatin and paclitaxel in first-line squamous lung cancer patients. The squamous population represents approximately 30% of lung cancer patients, and they tend to respond less well to checkpoint inhibition. The top-line data showed statistically significant improvement in progression-free survival, but no improvement in overall survival for this interim analysis. So that overall survival data is not yet mature. At ASCO, we are expecting the first numerical progression-free survival results as well as safety results. The positive progression-free survival results are a positive indicator for future overall survival results, which will be needed for approval. If approved, Centric is positioned to be the second PD-1, PD-L1 inhibitor approved for first-line lung cancer behind Keytruda. Merck will also be presenting data for Keytruda combined with chemotherapy for patients with squamous lung cancer. Earlier this month, Merck announced that it submitted a supplemental BLA for Keytruda in combination with carboplatin as first-line treatment for squamous lung cancer, with an FDA decision expected by March 3rd, 2019. This supplemental BLA is based on Keynote 407, and in ASCO abstract number 105, Merck provided detailed overall response rates from Keynote 407, and progression-free survival results are expected at the conference. At ASCO, we anticipate that the progression-free survival results from Empower 131 and Key 407 will be closely compared. Moving from squamous lung cancer to non-squamous lung cancer, we turn our attention to Empower 150, which is evaluating a triple combination of Centric plus Avastin plus carboplatin in first-line lung cancer. Detailed data for one of the primary endpoints, progression-free survival, were announced at AACR, and the abstract submitted to ASCO provides the first detailed data for the co-primary endpoint, overall survival, which are shown in this table here. Interestingly, the greatest benefit was seen in patients with EGFR or ALK mutations and showed a hazard ratio of 0.54. No such survival benefit was reported for Keytruda. The Keytruda trials were evaluating monotherapy or combination with the Keytruda trials, whether evaluating monotherapy or combination with chemotherapy, excluded patients with such mutations. This surprising finding will help differentiate Centric from Keytruda. A survival benefit was also seen in patients with liver metastases, but that had also been seen for Keytruda. Finally, we note that a survival advantage was seen in patients with PD-L1 expression, both above and below 1%. PD-L1 was measured in both tumor or interstitial cells, with TC123 or IC123 corresponding to PD-L1 expressed in over 1% of these cells. For these patients, the hazard ratio was 0.77. The TC0 and IC0 had PD-L1 levels expressed in less than 1% of cells and reported a hazard ratio of 0.082. This survival advantage should assure a wide label if approved. Speaking of approval, earlier this month, Genatech announced that the FDA accepted the company's supplemental BLA and granted it priority review with a PDUFA date of September 5th, 2018. With these positive overall survival and progression-free survival results, we are confident that it will be approved. So I'll finish this talk about checkpoint inhibitors in lung cancer with this summary slide, which summarizes data for Centric and Keytruda in first-line lung cancer. In the left column, we have data for squamous lung cancer, while on the right, we have data for non-squamous lung cancer. The Empower 131 presentation will have numerical progression-free survival data for the Centric chemo combination, which will be compared to data from Keynote 407. On the right, the we see the IMpower 150 presentation will be compared to data from the Keynote 189 that was presented at AACR. And finally, there will be a lot of attention on the late-breaking presentation for Keynote 042, which will present numerical overall survival results for Keytruda monotherapy in patients with low levels of PD-L1. Back to you, Mary Jo.
Thanks, David. It's a fantastic resource. So outside of immuno-oncology and first-line lung cancer, it looks like one of the targeted therapy fields, EGFR, could see an upset out of ASCO. David, could you fill us in on the data being released on Pfizer's Datamonitor?
Sure. So the final lung cancer presentation highlighted is for Pfizer's Datamonitor, a second-generation EGFR tyrosine kinase inhibitor. The trial, Archer 1050, compared Datamonitor to another tyrosine kinase inhibitor, gefitinib, which is also known as Iressa, in first-line EGFR-mutated non-small cell lung cancer. Importantly, patients with CNS metastases were excluded. In the table above, we see a 7-month improvement in overall survival for Datamonitor compared to Iressa, with a hazard ratio of 0.76. These data should assure regulatory approval of Datamonitor and help it compete against Iressa. Pfizer recently announced that the FDA accepted its NDA for first-line treatment of NSCLC in patients with EGFR activating mutations. The NDA was granted a priority review, and a decision is expected by September. Another tyrosine kinase inhibitor, Tagrisso, had its supplemental NDA for the same indication approved back in April. Tagrisso will be hard to beat, as it has a more favorable safety profile and has demonstrated activity in patients with CNS metastases. And that's all for me for lung cancer. Back to you, Mary Jo.
Thanks, David. Last year, Loxo Pharmaceuticals was one of the highlights of ASCO with its tissue-agnostic approach for its TRK inhibitor, larotrectinib. And this year, the company is presenting the second drug out of its pipeline, the RET inhibitor Loxo 292, which is similarly aimed at patients with RET alterations in this case, regardless of the tumor type. Zach, can you tell us more about that program?
Sure, Mary Jo. So as mentioned, Loxo Oncology's Loxo 292 is a potent, highly selective rearranged during transfection kinase, or RET inhibitor. In this phase 1 study, we have grouped here with the lung cancer information, even though it is being tested in multiple tumor types, because one of the main tumor types that it is targeting is non-small cell lung cancer. And from the abstract in the trial, they enrolled RET fusion positive non-small cell lung cancer and papillary thyroid cancer patients, as well as RET mutant medullary thyroid cancer patients and other cancers with these alterations. So from the data set, we got an ORR in evaluable RET fusion positive patients overall at 69%, 65% in non-small cell lung cancer, and notably 83% in papillary thyroid cancer. Tumor reduction was likewise achieved in 79% of medullary thyroid cancer patients, including 2 partial responses. And on the tolerability front, no dose-limiting toxicities were observed, the maximum tolerated dose was not reached, and no grade 3 or greater adverse events were attributed to Loxo treatment. So these results here are generally positive, and they show much improved efficacy compared to historical response rates with multi-kinase inhibitors. There are several multi-kinase inhibitors with at least some RET activity. Some of these include cabozantinib, vantetinib, and lenvatinib, as well as others such as sunitinib, sorafenib, alectinib. The list goes on. However, these multi-kinase inhibitors have not shown overall great clinical utility insofar as RET-positive specific patients. For example, lenvatinib in RET-rearranged lung cancers had an overall response rate of 16%, and in a Japanese study of vandetanib in patients with advanced RET-rearranged lung cancers, an objective response rate of 53% was reached in assessable patients. So notably, this data also reads through negatively to Blueprint Medicine's RET inhibitor BLU-667, which had previously disclosed fairly strong response rates in similar patients. So from this table here, you can see that the data from the phase 1 ARROW study of blue show clinical activity, albeit less so than LOXO-292, and more in line with those multi-kinase inhibitors I previously mentioned. You can see an ORR rates of 50% in RET-altered non-small cell lung cancer patients, as well as only 40% in RET-altered MTC patients. Now, RET fusions have been identified in approximately 2% of non-small cell lung cancer patients, and although that doesn't seem like a lot, the size of the non-small cell lung cancer patient population does mean there is commercial opportunity for RET-positive specific populations. In the U.S. alone, Datamonitor Healthcare estimates there are approximately 280,000 prevalent cases of non-small cell lung cancer in 2018. Just a back-of-the-napkin calculation there, 2% of that gives you somewhere in the neighborhood of 6,000 patients. So there is commercial opportunity in these RET-positive specific populations. In addition to non-small cell lung cancer, approximately 10 to 20% of papillary thyroid cancers, some subsets of colon cancer and gastric cancers have RET mutations, and RET point mutations also account for up to 60% of medullary thyroid cancer. So as mentioned, RET-specific agents do have potential market opportunity. RET rearrangements in non-small cell lung cancer specifically are commonly found in younger, as well as never or former light smokers, as well as in patients with lung adenocarcinomas, many of which are not differentiated. So even though RET mutations are less common than other lung cancer mutations, they can be successfully targeted, and efficacy in this population may improve with these RET-specific inhibitors over the multi-kinase inhibitors we currently have in the market. So looking forward to the ASCO presentation, which will be on Saturday. The presentation will utilize an April 2018 data cutoff date. The abstract presented here it uses a January cutoff date. So we're looking forward to an update there. The company has already disclosed that the efficacy has improved over time, so we look forward to that. And additionally, this abstract has also been selected for the Best of ASCO program, promising some good data.
Great, thanks, Beth. Turning from sort of lung and other to breast cancer, Zach, can you what are the developments to watch for in this space?
Yeah, sure. I'll keep things rolling here into breast cancer. You can see on your screen. One of the things that I wanted to highlight was a poster that may or may not have flown under the radar for some of the larger announcements here at ASCO 2018. And this is for G1 Therapeutics, G1T38, which is an oral cyclin-dependent kinase 4/6 inhibitor, CDK4/6 as they're commonly known. And this is from a phase 1B study. Interestingly here, the 20% partial response rate from the abstract and the 57% CBR are somewhat underwhelming. But again, I wanted to note that the primary objectives here were dose finding, safety, and tolerability, so that the fact that we have any sort of efficacy demonstrated this early on, even though it is not numerically what we like to see potentially compared to other CDK4/6 inhibitors, there's a hint of efficacy. But really the reason for highlighting it here for the ASCO presentation is its potential best-in-class CDK4/6 tolerability profile. So from this study of G1T38, patients dosed, there were no reported instances of VTE or QT interval prolongation, which are hallmarks of Verzenio and Kisqali treatment respectively. So for Eli Lilly's Verzenio, in the pivotal phase 3 Monarch 2 trial, VTE was reported in 5% of patients treated with the combination of Verzenio and fulvestrant, as compared to less than 1% of patients treated with a placebo and fulvestrant. And then as far as Kisqali is the only CDK4/6 inhibitor that has shown to lengthen the QT interval. In the Mona Lisa 2 trial, phase 3 pivotal study, one patient had a greater than 500 millisecond post-baseline QTc value, and 9 patients, or approximately 3%, had a greater than 60 millisecond increase from baseline. Moving on, G1T38 is associated with less gastrointestinal events, specifically diarrhea, than Eli Lilly's Verzenio. Verzenio is the only CDK4/6 which carries a warning in its label for diarrhea. Diarrhea occurred in 86% of patients receiving the Verzenio fulvestrant combination in the Monarch 2 trial, and as many as 90% of patients receiving Verzenio as a monotherapy in the Monarch 1 trial experienced all-grade diarrhea. Grade 3 or greater diarrhea occurred in 13% of patients receiving the combination in Monarch 2, and 20% of patients receiving the Verzenio monotherapy in Monarch 1. The next AE I wanted to highlight here was neutropenia, which is a class effect for CDK4/6 inhibitors. The 79% all-grade rate seen with G1T38 is similar to the approved CDK regimens. This is more in line with Ibrance and Kisqali treatment. Verzenio is the notable exception, as lower rates of neutropenia in approximately 46% of patients in the Monarch 2 trial using the combination Verzenio and fulvestrant experienced all-grade neutropenia, and 37% experienced neutropenia with the Verzenio monotherapy treatment in Monarch 1. This compared to Ibrance and Kisqali, which are more in line with the G1T38 results shown here. In Kisqali's case, neutropenia was the most frequently reported adverse reaction, with 75% of patients experiencing all-grade neutropenia, as well as grade 3 or 4 decreases in neutrophil counts reported in 60% of patients. As far as Ibrance is concerned, neutropenia was also the most frequently reported adverse reaction, with incidences as high as 80 and 83% in pivotal trials. So the neutropenia rate for G1T38 here is in line with approved regimens. And finally, perhaps the most noteworthy part of this poster presentation that will be presented is the ANC plateau here, allowed for continuous administration. This is currently only available with Verzenio. So in the trial, you can see that with both different doses, the 200 milligram and 500 milligram G1T38 doses, the ANC change at week 5 did decline about 50 and 75% respectively, but did plateau. Patients were able to receive G1T38 on a continuous basis, which would differentiate it from approved regimens with Ibrance and Kisqali, which require a dose holiday of 1 week per 1 month of treatment. So the continuous dosing, which is available with Verzenio, partly due to that lower neutropenia rate, would be an advantage. Now, on the flip side of that, this being a phase 1b study, development certainly lags well behind the CDK4/6 inhibitors that are approved, obviously. But if these results, as well as improved efficacy results, can be replicated in later phase trials, G1 Therapeutics, G1T38 CDK does have potential in this indication, specifically HR-positive, HER2-negative breast cancer, where these CDK inhibitors are approved, especially considering that the size of the market is fairly huge, and the CDK4/6 inhibitors alone have multi-blockbuster potential, specifically in Ibrance's case, that's already being realized. Now, moving on from the CDK4/6 inhibitors, the next abstract that I wanted to highlight focuses on PI3K inhibition. So this is a late-breaking abstract, LVA 1006, focuses on the phase 3 Sandpiper study of tacilicib and fulvestrant in estrogen receptor-positive, PIK3CA mutant advanced breast cancer patients. So Roche's tacilicib is a P110 alpha isoform specific PI3K inhibitor. This differentiates it from Novartis's pan-PI3K inhibitor buparlisib, which you may or may not remember was recently discontinued in all indications due to unacceptable liver toxicity in the phase 3 BEL-2 trial. So Novartis and Roche, which both have PI3K alpha specific inhibitors in development currently, are hoping that specificity can differentiate it from pan-PI3K inhibitors, which have had some difficulty in clinical development due to their respective tolerability profiles. So what we're expecting from the Sandpiper trial in this late-breaking abstract is the first quantitative results from this pivotal trial will be presented at the meeting, and this may lead to tacilicib's approval as the first PI3K inhibitor in this indication. And PI3K inhibitors generally are the next large drug class poised to reach the HR-positive, HER2-negative breast cancer market following the advent of CDK4/6 inhibition, which have become standard treatments. So as far as a market potential for tacilicib and alpelisib, which is Novartis's PI3K that's also in development currently, as far as the market sizing here, mutations in PIK3CA are present in up to about half of estrogen receptor-positive breast cancers. And for the time being, these inhibitors are likely limited to the PI3K pathway mutated patients. For example, top-line data for tazemetostat in a phase 1/2 trial demonstrated an overall response rate of about 38% of patients with mutated PIK3CA, compared with only 9% in wild-type PIK3CA patients. But this still represents a significant market opportunity given the size of the breast cancer population and as well as the prevalence of these PIK3CA mutations. In addition to PIK3CA, less common mutations in the pathway occur in things like the PTEN gene at a prevalence of around 2 to 4%, the RAC alpha serine threonine protein kinase around 2 to 3% as well. So there is potential for approximately half of patients with HR-positive HER2-negative breast cancer to be susceptible to PI3K inhibition. So we're looking forward to progression-free survival data at the presentation, as well as tolerability data, which is always important for PI3K pathway inhibitors. Further studies will really help us slot tazemetostat into the market if this data does read out positively for tazemetostat. For example, alpelisib, Novartis's PI3K alpha specific inhibitor, is also being trialed in the phase 2 BYLieve trial with fulvestrant or letrozole in patients with PIK3CA mutations that have failed both aromatase inhibition and CDK4/6 inhibition. So currently, we don't really know if these PI3K alpha inhibitors will be used as primary treatments or follow-ons to CDK4/6 inhibitors due to their standardization now within the treatment paradigm. But either way, they do have significant market opportunity. So again, this will be a late breaker presented on Sunday at the breast cancer session. We look forward to both the efficacy and safety data that will be presented there. Back to you, Mary Jo.
Thanks, Beck. And I'd like to thank all three of you. There's really been some excellent information and preparation on both the really high-profile presentations and some really interesting selections of what to monitor and why. As a reminder, please submit questions using the chat function. We have a couple minutes. And again, anything we don't get to today, the analyst can get back to you later. I'd also like to note that if you are interested in the pre-conference planner, that's available, and we'll be sending out a link so you can request any of our ASCO analyst reports, follow along with what we'll be putting out during the conference, request a pre-meeting planner. If you'd like, there will be an on-demand link for this presentation, and again, check out the planner, Scrip's review story, and keep your eyes out for lots more. And I think that really does bring us to the end of our time. We'd like to thank everyone for joining. Once again, keep your eyes out in your email. There will be a link for the on-demand recording of the presentation and the ability to request additional information that you'd like to see or find the rest of our ASCO content. Thanks very much.